Hello everyone. In this tutorial, we will learn to investigate genetic variants associated with complex trait diseases. As part of the investigation, first we will go over the GWAS catalog database to analyze the GWAS identified genetic variants that are associated with a complex disease. Second, we will use the odds ratio of disease-associated variants to estimate the relative risk of an individual for a complex trait disease, for example, Crohn's disease. Here is the GWAS catalog database website. It is a database of published genome-wide association studies and is maintained by NHGRI and EBI. Let's use Crohn's disease as an example to explore the database. A broader investigational question one might ask is, how many GWAS genetic variants have been associated with Crohn's disease? To answer this, let's start by searching the disease name on the search bar that will direct to a disease information page. The disease information page has 4 sections. The trait information section displays disease description. The association section lists all variants identified so far to be associated with Crohn's disease. For each variant, as indicated in each row, the table displays the SNP ID and the disease risk allele. In the first case, it's allele C for the SNP RS2249296, p-value of the association, allele frequency of the risk allele, it's 0.78 for the allele C in RS2249296. Odds ratio of the variant, it's 1.69 for the SNP RS2249296, indicating that the odds of disease risk in presence of the allele C is 1.69 that of the odds of disease risk in absence of this allele. The higher the odds ratio for a variant, the higher is the contribution to the disease risk. The mapped gene column indicates the genes to which the variant is mapped. In case of the SNP RS2249296, the variant is present in an intron of CLCA2 gene. However, because of linkage disequilibrium, other genes may be mentioned here. The next 2 columns indicate associated trait and link to the GWAS study details. The studies section lists all the GWAS studies carried out to identify variants associated with the disease. In the case of Crohn's, 40 studies have been carried out. For each study, as indicated in each row, the table displays author and publication details of the study. The discovery, sample number and ancestry column indicates the size and ancestry of the samples on which the GWAS was done. The replication sample number and ancestry column indicates the size and the ancestry of the samples on which the GWAS results were reproduced. The association count column indicates the total number of variants identified from the study that are significantly associated with the disease. The plot of catalog associations for selected trait across all studies section displays a Manhattan plot of all the variants indicated by each dot associated with the disease identified from various GWAS studies listed in the studies section. This is an interactive plot where clicking on the dot displays the variant information. In this case of Crohn's disease, the plot shows that the SNP RS5743293 in NOD2 gene is the most significantly associated SNP with the disease. Details on SNPs population frequencies can be obtained using databases such as dbSNP from NCBI and NOMADE from Broad Institute. Now that we have some understanding of GWAS SNPs and their properties, let's try to explore how one can estimate an individual's relative risk for a complex disease given the GWAS variants present in that individual. I will demonstrate this approach to estimate Crohn's disease risk. Crohn's disease is a type of inflammatory bowel disease that may affect parts of the gastrointestinal tract from mouth to anus. As shown in the figure on the left, there are many sub-processes such as barrier integrity of intestinal epithelial cells, inflammation, and activation of the innate and adaptive immune response are involved in the manifestation of the disease. Using GWAS, a number of Crohn's disease associated loci have been identified that are mapped to genes involved in these sub-processes. The table on the right displays a subset of the GWAS loci under each sub-processes. For each loci, odds ratio is computed based on case control data. These numbers indicate in some sense the contribution of each loci to the overall disease risk. An odds ratio value greater than 1 indicates a positive contribution towards risk, and a value less than 1 indicates a negative or protective contribution towards risk. Let's use this table to compute disease risk for an individual. Usually, in an individual, only a subset of the Crohn's disease loci are present. One can then compute the disease risk relative to an individual carrying reference allele for the loci. Under this assumption, relative risk can be computed as the product of the odds ratio of the loci present in an individual. In case of a dummy individual shown in the slide who is carrying 12 Crohn's loci, one can compute the risk contributed from each sub-processes by multiplying the odds ratio of the loci under each sub-process. Finally, an overall relative risk is a product of risk propagated from each sub-process, and in this case, it is a very high value of 4.66. However, in real life, the relative risk values of an individual for different complex trait diseases looks as shown in the table. As you can see, the individual has a higher risk for atrial fibrillation and protective for type 1 diabetes. This brings us to the end of this tutorial, where we learned to analyze GWAS genetic variants with respect to complex trait disease, and further learned a simple model to compute relative disease risk. Thank you for watching the tutorial.